Morning, everybody. Thank you very much, Phil, for the introduction, and thank you for collaborating with the UK Pharmacogenetic Stratified Medicine Network. So we've had 2 meetings now with the CRN and 2 with primary care, which is great. So I was asked to do something about potential genomics research in primary care and use warfarin as a case study. I will mostly focus on warfarin, but I will sort of talk about 2 other things at the end. Now, I'm going to take you in the next 25 minutes from discovery right through to implementation. So, the NHS spends £16 billion per annum on drugs. It's the second most important expenditure for the NHS after staffing costs. And as you can see, that the costs in terms of if you split it into primary and secondary care, most of the prescribing goes on in primary care, and that is still the case. But what is interesting is that the more expensive drugs are actually, and the costs in hospitals have actually been increasing in terms of, because more expensive drugs are actually prescribed often and started in hospital. They may be then continued in primary care as part of shared care arrangements, but overall you can see that primary care is still the most important area for prescribing. And this is an area, an opportunity in terms of pharmacogenomics or other aspects of personalized medicine where one can really make a difference and a potential gold mine in terms of undertaking studies, given 300 million consultations. I didn't know that figure. That's impressive. So warfarin, why is warfarin important? It is still the most widely used anticoagulant in the UK, 1% of the UK population. The problem with warfarin is that there's a huge deal of variability in the dose requirement per day, 40-fold. Some patients will require half a milligram a day. Some patients require 20 milligrams a day. And we have a biomarker called INI, International Normalized Ratio, that we use. We try to keep it between 2 to 3 for patients with atrial fibrillation or DVT thromboembolism, pulmonary embolism. But we often get it wrong. And when we get it wrong, particularly when the INR climbs above 3 or 4, you get bleeding, as in this 82-year-old patient with a calf hematoma. And obviously, when bleeding occurs, as it did in this patient, it may cause mortality. But in this lady, she was in hospital for 6 weeks with us afterwards, recovering from the hematoma, trying to walk again, learning how to walk again, and so on. So there's huge consequences of getting things wrong. So I'm just going to give you some pharmacology. I am sorry it's early in the morning, but I am a clinical pharmacologist, so I have to do that. So warfarin is administered as a racemate. It's an R and S enantiomer. The S enantiomer is 3 times more potent than the R enantiomer, and that's largely, not completely, responsible for the anticoagulant effect of warfarin. The cytochrome P452C9, which is a major P450 involved in metabolism of the S enantiomer, is polymorphically expressed. And there are many different polymorphisms there. The commonest ones in the UK population are something called STAR2, STAR3, which reduce the activity of cytochrome P452C9 by up to 90%, depending which variant you have. Warfarin interacts with vitamin K epoxide reductase complex, which is the enzyme involved in the vitamin K cycle, which leads to the formation of activated coagulation factors 2, 7, 9, 10, which are the vitamin K dependent clotting factors. And VCOC1, and the gene for that, is also polymorphically expressed. There are different haplotypes called H1, H2, and different people with different haplotypes have different differential levels of activity of VCOC1. So if you, theoretically, if you have low activity variants of CYP2C9 and VCOC1, then you're likely to require lower doses of warfarin. And certainly that is proven to be true in terms of discovery studies which have been done. And studies have been undertaken all over the world, and this is something that we did within UK, where we undertook recruitment of a prospective cohort. It was largely from secondary care, but also from primary care. We did sort of recruit from primary care patients who were on warfarin, started on warfarin, and then followed them through both secondary and primary care. For those of you who don't know, this is it's called the Manhattan plot. You have the chromosomes on the x-axis and the minus log 10 p-value on the y-axis. And what this shows is a million SNPs which were genotyped, and then another 6 million SNPs which were imputed into each individual. So each individual there has 7 million SNPs, which we've looked at and tried to define what determines the sort of dose, individual daily dose for warfarin. And although this is called the Manhattan plot, and it's called Manhattan plot because when people started working in complex disease areas, they got lots of skyscrapers, basically, lots of peaks coming up, and it looked like Manhattan, the Manhattan skyline. But here we've only got 2 skyscrapers, really, and so I don't know what city that fits in with, but it's not a Manhattan plot, but it's good for us to be able to get 2 genetic hits there with cytochrome P452C9 with a p-value of 10 to the minus 30, and VCOC1 with a p-value of 10 to the minus 50. And so these 2, if you then take into account what determines daily dose of warfarin, these 2 account for the largest degree of determination. Over 40% of the daily dose of warfarin can be determined by 2 genetic risk factors. There are obviously clinical factors that are important as well. Age is important. So as you get older, your warfarin dose requirement gets lower. And that's because your liver starts getting smaller as you get older. So that's something for you to look forward to. As one gets bigger, heavier, your warfarin dose requirements go up. And obviously there are interacting medications as well. In these days of polypharmacy, multimorbidity, many patients will be on interacting medications. So for us to be able to then take this into further practice and so on, and see how we can actually see where they can work in clinical practice, we had to undertake a randomized controlled trial. Before we could undertake a randomized controlled trial to see whether it was worthwhile genotyping patients for these particular polymorphisms, we had to develop an algorithm. So we worked with researchers all over the world, and we developed an algorithm which incorporated both the genetic factors I showed you before, as well as clinical factors such as age, body mass index, etc., and interacting medications. And this was published in the New England Journal of Medicine. And from this, then we decided to undertake a randomized controlled trial. Now, the warfarin dosing pathway, irrespective of whether you're in primary or secondary care, most anticoagulation clinics now are sort of becoming more community-based, is really shown here. Is that you have a clinical decision to start on warfarin or another anticoagulant. I'll come back to the other anticoagulants in a minute. Most places still use loading doses, usually 3 doses, but the total number of how you load varies quite a bit across the country. We've done a survey which shows that it does vary quite a bit across the country. On day 4, usually you have your INR checked, and then you're dosed either based on manual dosing or computerized software dosing. And then hopefully you're reaching your target INR, and you go into the maintenance dosing, and you actually start being dosed via computer systems, algorithms. There's Dawn, RAID, and various other types of systems which are available in primary care for dosing, for maintenance dosing of warfarin. So what we want to do is, if we were able to impose genetic dosing onto this, how would it fit in? Well, the first thing is that we would need to develop a loading dose algorithm, which is what we did. Then you'd have to develop an algorithm which took into account the fact that you're measuring an INR on day 4. Because some people may have a very high, marked increase in INR based on loading dose, others may not. And so you need to make sure that the dose you give on day 4 is based on, takes INR into account. And then finally, you can just use usual dosing software after day 5. So we developed a randomized controlled trial by using 2 algorithms and genetic dosing for the first 5 days, followed by usual dosing via software for the rest of the time. And we did this in the UK and Switzerland, and it was funded by the EU. And what we basically did was randomize individuals to the genotype guided dosing arm, or into standard care arm. And the standard care arm was 10 milligrams on day 1, 5 milligrams on day 2, and 5 milligrams on day 3, unless you were over 75 years old, which was 555 with the sort of dosing in the standard care arm. And we used a point-of-care test for genotyping, which was working with LGC, and it's good to see Simon Wells here from LGC. And LGC were part of the consortium for this EU consortium. This particular point-of-care test gave us a result in 2 hours within the trial. And patients would come in, if they were randomized to genotype-guided arm, they would have a blood test taken, it would be put into the machine, and it would give us the result within 2 hours. And then we used the dosing software algorithms, which are on the web, to be able to dose them for the first 3 days. And then they came back on day 4. And we published this again in the sort of New England Journal of Medicine. And what we were able to show was that genotype-guided dosing was better than standard care dosing. Let me just take you through this slide. Standard care dosing, if you look at the control arm, what we got at 3 months, and the primary outcome measure was time in therapeutic range within the first 3 months, we got 60% time in range within the first 3 months. Now, that is a fantastic result. You don't normally get that in any kind of usual anticoagulant clinic. This is because it was protocol-driven. We had very strict INR monitoring going on within a randomized control trial. So to get 60% is pretty good. But despite that very high INR within the first 3 months, we got 67% in the genotype-guided arm, which is 7% difference now, which is highly statistically significant. When you actually start stratifying it to those who have the variant versus those who don't have the variant, those who have 2 or more variants in those 2 genes I showed you had an 11% difference in time in range. Now, people say 11% isn't very much. Actually, that is clinically relevant. Studies have shown that a 10% improvement in your time in therapeutic range leads to 20% improvement in clinical outcomes. So this is a clinically relevant change. Now, people said, well, why didn't you do a study where you had clinical outcome measures? Well, that would require a trial of 18,000 patients. Now, nobody is going to fund me for an 18,000 patient trial for Warfarin at the moment. So we had to use INR as a surrogate there. This shows you the Kaplan-Meier plots. And what you can see here is that individuals who were in the control group were much more likely to overshoot, while those who were in the genotype-guided group, which were being dosed via the algorithms, were less likely to remain at 2.5 and remain within the range. And again, also in time in therapeutic range, you can see separation occurring between the genotype-guided group and the control group at about 10 days. And it was still present, that separation, at about 3 months, which was our sort of primary outcome measure. Now, at the same time, in the same journal was the American trial, which was published, which was called coag. And although we showed difference between genotype-guided dosing arm and the control arm, they showed no difference at all between genotype and control arms. So that led to a lot of controversy. We then produced a paper in Trends in Pharmacological Sciences saying why there was differences between the trials. And one of the major reasons was that the American trial had 28% African Americans, whereas ours was 97% Caucasian. The African Americans have different polymorphisms, which weren't actually tested for within the sort of algorithm that they were using. They only tested for the Caucasian polymorphism, STAR2, STAR3, and the VCOC1, which is exactly what we tested for as well. So that was one of the major reasons why it didn't work. Last week, another trial came out from the United States, and this trial, which has just been published in JAMA, actually confirms our results. And they found that there was a 27% reduction in the primary outcome measure, which was a composite of death, venous thromboembolism, overdose, etc. So again, now there are papers already appearing in the American literature saying it is time for us to rethink warfarin genotype-guided dosing. We never stopped thinking about it, and we carried on. So one of the things that you have to do to be able to start thinking about implementation is to think about cost-effectiveness, because obviously we live in resource-constrained times, and you can't start putting in very expensive tests into NHS without knowing what the cost-effectiveness is. And what we were able to show was that actually this was highly cost-effective. This is the UK curve. And ICER, incremental cost-effectiveness ratio, was £6,700. As you know, the threshold in the UK is between £20,000 to £30,000 that NICE utilises, and so this was highly cost-effective. In Sweden, it was less cost-effective, but nevertheless still cost-effective according to their threshold. It was 250,000 Swedish krona, and their threshold is about 500,000 Swedish krona. Now you may say, why is it less cost-effective in Sweden than in the UK? Well, Sweden is #1 in terms of quality of anticoagulation, irrespective of whether you use genotyping or not, and the UK in the league table is #5. So that's why it was less cost-effective, but it is still cost-effective in Sweden. So what we wanted to do next was that it was randomized control trial. People say, well, if it's randomized control trial, we don't believe it's going to work in real clinical practice. So how can you actually then take it into an implementation? And this is where we did start working with community-based clinics and primary care type clinics. So we initially said we want to do implementation study. And we had a discussion with the ethics committee who said that this was more a service evaluation. You've already done a randomized control trial, so you don't actually need ethics, but you need some kind of approval from the innovation departments. And this is where our problem started. We needed to find out how we get these kind of approvals. And there's no gold standard, actually, to follow within the NHS in terms of how you get approval for innovation and undertaking a proper evaluation of that. And the processes tend to be different in 3 trusts. And so approval for this innovation took more than 6 months to be able to take through. And there was lack of clarity about some of the information governance processes, what we were allowed to take out, what we were allowed not to take out in terms of data and so on. And certainly, even when we wanted to get all the data dumped from all the sort of anticoagulation clinics within the Northwest Coast in terms of what the control was in terms of INR, we weren't actually allowed to do that even when anonymized. We managed to get some dashboard data in the end, but a limited amount of data. We also worked with LGC, and they had refined their genotyping equipment, and they had a new platform called ParaDNA. And we undertook some rigorous testing at the university to identify numbers of ambiguous results, why they were occurring, and then worked with LGC to be able to get this as good a quality possible of this genotyping that could be utilized in clinics by nurses, etc. They also were very good in coming to us and giving training to the nurses in the community-based clinics who were actually going to utilize the particular platform to undertake the genotyping. Obviously, if you were going to introduce a new platform in a clinic and so on, you need approval from various different departments, medical engineering, etc., and also some standard operating procedures of how to be able to utilize the equipment and clean it and so on properly. One of the things that we found early on was that when we wanted to go to community-based clinics, they didn't actually have a freezer for storage of kits and so on. So we had to buy some things like that. So although you may think that undertaking a randomized controlled or implementation study may be related to other kind of difficulties, actually it was something as simple as not having any kind of storage facilities to be able to take through the implementation. So this is the ParaDNA point-of-care device. And basically you just take a mouse swab, you put it into the wells you can see over here, you put it then into this sort of machine, and you get a result now in 45 minutes. So it's gone down from 2 hours to 45 minutes, which is fantastic. So the patient can be waiting for the result while the sample is being analyzed. And it gives you a result like that, and the nurses were able to use it, and they were then able to put it into the algorithm on the web and be able to dose the patients as previously described. And here's some of the nurses who've been involved in this, and this is one of the patients who was given consent to be there, and he was basically very happy to be able to take part in it. Unfortunately, he was case of 1, n equals 1, who said it worked for me, and my INR got into the right range at the right time, unlike his mother, who had lots of difficulties with warfarin. But however, this has led to some publicity for the fact that nurses are using in primary care clinics a gene test to personalize warfarin dose. For example, there have been various articles, including in the Nursing Times and so on. And it's been very much nurse-led, and not GP-led, not hospital physician-led. It has been very much nurse-led to be able to do this implementation. And the nurses have worked very hard to be able to do this, so which is fantastic. So what we've tried to do is to go from a randomized control trial to implementation. And the implementation is completed now. We haven't fully analyzed it, so I can't show you all the data. But what we're finding is that we're getting exactly the same result as in the randomized control trial. In the randomized control trial, we found there was a 7% difference in favor of genotyping. That's exactly what we're finding in the implementation study. So in a real-world setting, etc., we can actually show the same things that happened in a randomized control trial. So basically, what's the point of doing this? There are lots of new oral anticoagulants out there. Rivaroxaban, apixaban, edoxaban, and dabigatran. This one is not licensed yet. Betrixaban is not licensed yet. And clearly, these are now being used. They're marketed on the basis that you don't need any kind of monitoring. And patients obviously don't like coming to anticoagulant clinics to have their monitoring done. And to some extent, doctors don't either. You know, it's clinician choice as well as patient choice. So these are a great innovation. However, there are issues associated with these oral anticoagulants. This is a paper which has come out recently from Canada in a real-world setting. And what they were able to show with rivaroxaban and apixaban was a 60 and 50-fold variation, respectively, in plasma concentrations of those two drugs. And 12 and 13% of patients on rivaroxaban and apixaban, respectively, exceeded the 95th percentile for maximum plasma concentration. So that puts these patients at risk of bleeding from these particular drugs. We don't monitor them at the moment. We don't know what is going on, and we're doing some work in a UK-based setting to see what's going on with rates of bleeding and so on with these particular drugs. Another worrying thing, and I'm grateful to John Byrne for providing with this slide, is that adherence data for oral anticoagulants. You can see this is a 12-month look at in 2016 and from general practices. And what they find is that at 12 months, the adherence rate with warfarin is greater than 80%. With dabigatran is getting to 40%, and with apixaban and rivaroxaban it's about 60%. And basically what may be happening is that patients are not being monitored, therefore they don't take the drugs because nobody's monitoring them, and therefore adherence may lead to problems long-term in terms of lack of efficacy in these particular patients. So there are issues that we need to be able to contend with. Now, there is also a cost issue as well for DOACs in the NHS. So if you look at the fact that the patents are going to expire in 2023, the total increase in prescribing is 504% for apixaban in 2013-14. And I think in 2016, last year was £300 million spent. This year, or maybe next year, it will reach 1 billion, which is 5.5% of the total NHS drug budget, which is more than the Cancer Drugs Fund, I think. Okay, so that's going to bankrupt the NHS. And we could still be utilizing, actually, DOACs are a great innovation, don't get me wrong. You know, there certainly are sort of an important group of new drugs, but it is possible that we could stratify, and this is where stratified medicine actually works, is that you could actually genotype individuals, and if they haven't got a variant, and 67% of you in this room won't have a variant, then you could be given warfarin. I know from my time on the wards that whenever I went home at the weekends and I came back on a Monday, there were a small number of patients on warfarin who were really badly dosed and were all over the place by sort of, there's people who were on call over the weekend. But the majority of people, no matter how bad you dose them, actually remain quite stable within the INR range. And that's, I think, is the 67% of people, and we've shown that in a prospective cohort study. And so my feeling would be that we could easily save NHS England £500 million by just actually stratifying to warfarin and DOACs. So Now, another example I want to show you is about clopidogrel. Now, clopidogrel is used in coronary artery disease, but also in cerebrovascular disease. It is an antiplatelet drug. It is a prodrug which requires to be metabolized to active metabolites, which then work on the receptor on the platelets to be able to cause platelet inhibition. The major P450 enzyme responsible for metabolism of this is cytochrome P450 2C19. Now, this is also polymorphically expressed. And people have shown in very high impact papers in big journals that if you have a loss of function polymorphism, then you're much more likely to get stent thrombosis, for example. You can see the STARS2 is homozygous for the loss of function variant. And heterozygous are also at increased risk. And this was with stent thrombosis. Another paper from New England Journal of Medicine from Jessica Megger again showed with this Kaplan-Meier plot that those individuals who are actually carriers are much more likely to get stent thrombosis and so on after receiving clopidogrel. And people have started developing rapid gene tests as well, again, point-of-care tests. And this was a proof-of-concept trial undertaken in Canada. It was actually done in a coronary angiography lab, not in primary care base, but again, you should be able to utilize this in primary care. It was done by nurses, and basically what they were able to do was to genotype individuals, and randomized to genotyping was the standard treatment arm. And when they did do it via genotyping and use an alternative drug in those who had the variants, they were able to show they got better platelet inhibition with the genotype-guided dosing compared to standard treatment. There's been many studies now which have shown that using clopidogrel and without genotyping actually increases the risk of ischemic strokes as well as other types of cardiovascular events. But at the moment, we don't use this at all in the NHS. There are trials which are going on in the United States at the moment to be able to look at this. So just to finish off, I have focused on pharmacogenomics. Obviously, there are many other types of things you can look at in terms of genomic research. There are different models in terms of how primary care can contribute to genomic research. It can be from collection of samples to more complex scenarios, such as the Warfarin example I've given you, where you use algorithms. It can be nurse-driven, etc. Now, we have done this collection of samples, and we did this particular study using funding from the Wellcome Trust and MRC, using health record data. We worked with CPRD. We were able to link CPRD records to biological samples. And we used statin myopathy as the phenotype. And from that, we were able to collect 250 patients with statin myopathy, and were able to show this particular variant was associated with simvastatin myopathy. We've now done exome sequencing on those patients, and that data is just being analyzed. So you can actually undertake just sample collection, working with GPs and so on, and being able to identify patients using the sort of big data examples and so on, big data approaches that Phil talked about earlier. Obviously, for more complex tests, it would be difficult to implement them within primary care, but they need to be done in labs, but they can still be patient recruitments, they can still actually go on. I think an area which would be interesting to hear from the GP colleagues here is point-of-care genotyping. Obviously, there are constraints of time, costs, and so on, which need to be assessed within primary care. Obviously, capacity and capability and training issues, which were mentioned earlier, also need to be looked at in terms of undertaking this kind of research. So thank you very much for your attention.